I haven't had a chance to look at the homeworks yet, but I'll try to do it. I don't know. Try to do it today or early tomorrow, and I'll get back to you whether I'm doing homework or extra credit. I've also been looking at. I realize there are actually 9 lecture sections for this part of the test, which is more than usual. So I was thinking maybe I might just finish up lecturing on Tuesday and then running a review session in class on Thursday. How does that sound? Does that sound okay? So the exam is coming up a week from Tuesday, so I'll try to get a study guide out. I'll see how the material goes. I might have to do a little bit of lecturing on Thursday, but I don't think I'm gonna skip I'll do the evolution in medicine thing, but I'm gonna do the phylogenetic tree reconstruction after this lecture today. So, and that'll get you prepared for another lab. And I'll probably have a homework associated with that, so I'll give you a little experience how to build phylogenetic trees for your lab. Anything else? I don't think there's anything else. So what we'll do today is we're going to finish up on coevolution. And what we've been focusing on are these antagonistic, these negative interactions between, we were looking at predator-prey interactions, and now we're going to look at host-parasite interactions. And this kind of gets at the question of what we call the evolution of parasite virulence. And does virulence mean anything to you guys? Is that a term that you've ever heard? Yeah, well, you hear about it like it's kind of related to a virus. Viruses can be virulent, but virulence is kind of how negative a pathogen or a parasite is on a host. An extremely virulent one has really negative consequences, often leading to host mortality or host deaths. Some can be kind of intermediate virulence, having little effect on host conditions or fitness, and then some can be avirulent. And so what we're going to look at, we're going to use an example of an interaction between a virus and rabbits that was used in a particular contest to look at how parasite virulence can evolve. And what do you think you guys are all hosts, right? Some of you are experiencing some attacks by some pathogens probably right now. I hear some people sniffling, right? Okay. So we're hosts. We have a host-centered view of the world, right? And where do you think pathogens should go? What's the most successful pathogens? Ones that should be have little negative effect on their host, do you think? Yeah, you might think. And why might you think that? Yeah, in some general way, having a really negative effect on your host and killing it, you're killing the thing in which you live, right? So you might think in some ways that's probably not a good strategy to maximize your fitness as a parasite, okay? And that in general is true, but often people have been led to believe that parasites should evolve to become relatively avirulent because they want to have a minimal effect on the host. Well, that isn't often what we see, and it depends on how. So one of the things we're going to look at, I want you to think like a parasite, right? Get out of your host-centered view of the world and start thinking about a parasite. And from a parasite's perspective, and we'll show you an example of this, from a parasite point of view, selection favors individuals that maximize their transmission to new hosts, right? What do parasites want to do, right? They don't want to stay inside you. They want to get transmitted to infect another host, right? That's all part of the parasitic life cycle. Whether you look at any sort of parasite, it's host-to-host transmission, which is critical to maintain sort of populations and those sorts of things. So we'll start with the simple premise that selection favors individuals that maximize transmission to new hosts, okay? Now, the idea here is if you're in a host, right, what do most parasites do inside hosts? And what are some of your bacteria or viruses doing inside you? They're replicating, right? They're reproducing, okay? So you can often get these parasites can have often negative effects on hosts because parasites often multiply within hosts. They're reproducing, they're using host resources to replicate and reproduce, right? And prior to the releasing particles to potential new hosts. What we're going to look at is a kind of a cool example that illustrates how virulence can evolve and how it doesn't often go to really low levels of virulence. Often intermediate levels of virulence can be favored by natural selection. And so we're going to look at a particular example of a thing called a myxoma virus that's transmitted between rabbits via a mosquito vector, okay? And usually where we see parasites having really strong effects on particular host fitness is when they require another species to transmit the host. So a lot of insect vectors that are needed to get this myxoma virus can't be directly transmitted from one rabbit to another. It has to be transmitted via mosquito vector. Okay? And how do you think those mosquitoes transmit the parasite from an infected rabbit to an uninfected rabbit? What do you think is the mechanism? Huh? Yeah, when it goes to feed on a different rabbit, these are probably blood-sucking mosquitoes, right? Like all of them are, right? And that's how they transmit that particular virus between an infected host and an uninfected host. Okay, so what we're going to look at is how this myxoma virus is transmitted between the rabbits via mosquito vector, and then sort of what we're going to talk about is different levels of reproduction of that virus within rabbits often has different effects on host mortality, right? So how do you think that's correlated? If you have a highly reproductive myxoma strain, what do you think the fitness effect is on the host? It'd be more virulent, okay? So usually there's a tight linkage between the level of replication of a virus or a bacteria, right, and its virulence. High levels of replication, high levels of reproduction within hosts often lead to high host mortality. If you have a virus or bacteria that doesn't replicate very much, right, is using very little host resources, right, during its growth, well, probably has minimal effect on the host, okay? So we're going to talk about, there's some trade-offs here in terms of how quickly you reproduce, how well you can be transmitted and that sort of thing, and we'll use this example. Okay. So this is one of the classic studies this is another one of these kind of classic studies of sort of host-parasite interactions that we all continue to talk about, even though it was done in the 50s and up through the 80s. So the reason we're going to focus primarily on what happened in Australia, and how do you think it went when rabbits were introduced to Australia? Do you think that was a good thing? Well, it turned out it was probably in about, I can't remember exactly when it happened, but I think it was in the early 1900s. They brought over 12 pairs of rabbits to Australia. Guess what happened? They got loose. And then what do rabbits do? They breed like rabbits. Okay, they got loose. And rabbits are not native to Australia, right? So Australia obviously has a lot of unique native plants and animals, and people got concerned because these rabbits went through these sort of exponential growth phase. They increased in numbers rapidly. Guess what the consequence was to sort of native plant and animal communities in Australia? It was bad. So rabbits are herbivores. They eat plants. They sort of decimated a lot of the communities. And so somebody had a great idea to use a biological control agent to reduce the population sizes of Australian rabbits. And guess what they introduced? The virus. Okay? I'm not going to go into all the details because it's kind of, this strain of virus was developed in Brazil in the 1950s, and they actually developed an extremely virulent strain of this myxoma virus. They then, I'm not sure this would not happen anymore, okay? Don't get me wrong, we probably wouldn't be doing this anymore. But what they did, they then introduced this extremely high level of virulent strain into the Australian rabbits, okay? Guess what happened? A lot of them died, okay? And then over time, we can look to see what happened. So what do you think should happen from both perspectives? Let's talk about what should happen in the rabbits that are being exposed to this extremely virulent virus. What would you expect to happen? Yeah, so the idea you would expect that, selection for resistance, genes in rabbits that are more resistant to the myxoma virus. There would be really strong selection for that. What do you think should happen to the virulence of the myxoma virus? It should decrease, okay? And both of those things happen, but let's talk about what happened. We're going to focus primarily on what happened in the rabbit. Okay. So this is we're just going to focus, we're not going to look at France and Great Britain and all these other things that, other studies that were, so, okay. So basically, the myxoma virus actually evolved lower virulence over time after its introduction in all these various places. And you can categorize the virus into sort of 5 virulence grades. 1 is the most virulent, 5 is the least virulent. And these are kind of intermediate virulent strains, okay? So in 1950 or 51, what did they introduce into the Australian rabbits? What kind of strain? Yeah, that was the one. That was the one that was brought over from Brazil. And all of the myxoma virus, 100% of the highly virulent strain, okay? We can skip over a lot of this stuff, but what eventually happened by 1963 and 1964? What's the outcome? You see none of the super virulent strain, right? So it was strongly selected against. So clearly that was bad, right? Selection eliminated it. Did we go to avirulence? Which is this one, this is an extremely avirulent strain that has really low replication rates. Did we get that? Now, basically what we have is the frequency of these strains, kind of 3A, 3B, and 4, and it depends on which populations you're in. But in general, selection led to these kind of intermediate virulent strains becoming more common, okay? Here's what happened to, and I'm not going to go into all the details, but unselected rabbits, they had strains of rabbits that had never been exposed to myxoma, and there was 100% mortality of those, and that's what happened early on. But then they took a bunch of rabbits after they'd been exposed to the rabbit, and they showed much lower mortality. So some resistance had evolved, not complete resistance, but these things showed not 100% mortality. These are rabbits that had been selected, that they came from the wild in Australia. They'd experienced some interactions with the myxoma virus, and they had lower mortality rates. So there appeared to be some ability to deal with the virus and some resistance, not completely though. So rabbits evolve resistance over time. Okay. So what we want to know here is, why were these selected against, right? Why do we also not see it going to avirulence, and why was intermediate virulence favored? Any ideas? What do you think it has to do with? Yep. So if it was completely avirulent, right, and think about who's spreading this virus. It's not the rabbits. It's a mosquito vector, okay? And where's that virus replicating, and where is it present in order to get transmitted from an infected rabbit to an uninfected rabbit? What's the part of the system of a rabbit that it has to get into? Into the blood system, right? And these are blood-sucking mosquitoes, right? So they have to get access, okay? So if you're extremely avirulent, what do you think the density is of viral particles in the bloodstream? They replicate it's really low, okay? If it's extremely virulent, well, I'll just show you what happened. And you don't want to see the pictures, okay? So the idea here is there were highly virulent strains, the grade 1s. What they do is they create these huge lesions on the rabbit. It kills off the host really quickly. The presence of all these viral particles and all these lesions and all these things results in really rapid host mortality. And the reason is, even though there's a lot of virus particle, even in the lesions, they can get access to it. The take-home message is these highly virulent strains have a really low transmission rate, because they have a kind of this short period for successful transmission by the vectors. They die too quickly. And this is that makes sense, doesn't it? There's really strong selection against high virulence. Low virulence is the animals recover quickly, right? And they actually have no open sores. Remember, rabbits have hair, right? What do mosquitoes like? They probably want access to skin, okay? And so they have no open sores for mosquitoes to get easy access to blood. So not only do they have really low densities of viral particles in the blood system, they don't produce any open sores. So it turns out this vector there's very little transmission from an infected rabbit with a low virulence strain to uninfected individuals. So there's a really low transmission rate. So both of these were kind of selected against. The intermediate virulence is mosquitoes easily bite. These had much smaller open sores, and mortality does occur, but it takes often 30 to 40 to 50 days for the mortality to occur, if it ever does at all. So the mosquitoes can easily bite smaller open sores, and the mortality takes longer. And it turns out these kinds of strains have the highest transmission rate. That is, they result in a higher rate of infection of new rabbit hosts. So the basic idea here is parasite virulence evolved from high virulence to intermediate virulence because that really maximized the transmission rate. It didn't go to avirulence because that had a really low transmission rate. So in this particular system, and this is true in a lot of host-parasite interactions, especially things that involve vectors, organisms that move a parasite from that host to another host, these are things that often have kind of intermediate levels of virulence, and the reason is it kind of maximizes the transmission rate. So they're not entirely harmless. They have they produce these open sores, and they have a high level of virus particles in the blood system, and they're still they have intermediate virulence. Okay. Does that make sense? Okay. So the take-home message. So from a perspective, from a parasite's perspective, it's not, oh, I don't want to harm my host, right? The idea is selection favors the level of virulence, how nasty it is to the host, that maximizes its transmission rate from an infected host to an uninfected host. Okay. Let me stop. Any questions about that? Does that make sense? Kind of? Okay. Let me stop this recording.